So we've now made it to the second part, which is glycogenolysis. So which is a particular part of the video a friend of mine requested for. So first of all, to understand this, you had to understand glycogenesis first. Again, if you go back to the old video, this is our end product here. This is a glycogen molecule, and again we said this is the reducing end where there's the one carbon, and this is the non-reducing side. And again, if we go back, we said that by the work of the branching enzyme, there will be a bond here, which is the 1, 2, 6 bond here, which is again alpha 1 to 6 glycosidic bond. Okay, so in this reaction, or in glycogenolysis, again, glycogenolysis simply meaning we're breaking down glycogen into the release of a ready form of energy, which is glucose. So there are 3 enzymes that I want us to look at that will perfectly explain this whole process. It's only 3. So the first one is glycogen phosphorylase. So when you think about this enzyme, the way I want you to think about it is the way I'm going to draw it. So this is the enzyme here, right? So this guy is a ninja, and you know ninjas are famous for attacking. When they're attacking, what do they throw? They throw shurikens. So for this instance, the shuriken this guy will be throwing will be a phosphate, right? But what will this phosphate do? And then again, this is where the importance of understanding the reducing and non-reducing sides. So when it's breaking down, it doesn't break down from the reducing end, but from the non-reducing end. So when this guy is done, so let me just show some lines to show that there's some velocity going on here. So when this guy is done here, when it breaks down this bond, what are we going to yield? So what we're going to yield in this case will be what is known as glucose 1 phosphate. And again, glucose 1 phosphate, if you look at the structure, so it means that the 1 phosphate will be attached at the number 1 carbon atom, which is here. So this is what this enzyme will do, and it will continue breaking them down from this non-reducing side. So when it breaks them down, this is the end product that we're having. So again, it's breaking from the non-reducing side though. So we're going to have here glucose 1 phosphate here, so phosphate here, phosphate here, phosphate here, phosphate here, phosphate here, here. Okay, I'm sure you get the idea. So again, all these glucose 1 phosphate. But then again, we don't want this, because we want a form of glucose that we can use for energy. And this is where this second enzyme will come in, which is phosphoglucomutase. So phosphoglucomutase, what it will do, it is okay, so I'm going to go into a little bit more into the mechanism. So this guy, what he has, basically the phosphoglucomutase is formed of 2 things. So it has what is known as a serine residue attached to a phosphate, right? So again, we have this glucose 1 phosphate. Let me just quickly roughly draw it. The glucose 1 phosphate here, so the phosphate is somewhere here, right? And this here is our phosphoglucomutase. So what does this guy do? So what this guy will do, basically in this step, so there's a carbon number 6 here, right? The carbon number 6, so it's attached to OH and H. Okay, so what this guy will do is that it will get this OH here, right? So it will be this serine residue like this attached to the OH. So what happens is that the phosphate will remain just outside. But what happens now is the part that's really important. So the thing is, once it loses this, it means it will have space to accommodate something here at carbon number 6. And what will it accommodate? The phosphate here. Okay, so we said again, the phosphoglucomutase, again, it has serine and phosphate. And then in the first step, it will, first of all, take the OH here from this guy here. But then again, since this is a very selfish guy, so what he will do next is that, again, instead of just leaving things as they are, he will come again, donate this OH here, and then again, this phosphate that it lost initially will come in here at carbon number 6. So what does that mean? So then this will be a new form. So again, quick sketch. So this will be the new form. So again, he's given this OH here now. So this will just be a normal carbon attachment. So carbon again to OH, OH, H down here, and then up here. The phosphate will go up here. So again, carbon number 6, there will be a phosphate attached. Very, very important here. So at this point, what molecule do we have? We will now have what is known as glucose 6 phosphate. So why is it important for us to have glucose 6 phosphate? So again, glucose 6 phosphate in the muscle cells, if you release the glycogen, glucose 6 phosphate will directly enter into a glycolytic pathway. So meaning it will now go on and yield ATP, which is a form of energy. And in the liver, what happens is sort of different. So in the liver, the function of the liver basically is that when you're not eating, you're in your fasting state, your body kind of wants energy back to itself. So what will the liver do? The liver will simply release glucose, not glucose 6 phosphate, but glucose back into the system. So once you yield this glucose 6 phosphate, there's a phosphorylase enzyme that will come in, again, cleave off this phosphate to just yield the free glucose. So this is basically what the phosphoglucose does, and the important implications in the whole process of, you need energy. There's this form that you can utilize, which is glycogen. But then again, it doesn't end. So we've said up until this point, it's cleaved, right? So these guys, all of them here, we just simply rub them off. They've gone. They're free. Some of them have been converted to glucose 6 phosphate, some of them into glucose normally by the liver, because you need energy at that point in time, you're fasting. So now I'm going to go into the next step. And again, I'm pretty sure you're wondering why this guy alone is the only one that's looking inside from his friends. So it is simple, because this guy will be the last one to be involved in the whole process. So there's another enzyme that comes here, a very, very important enzyme, which is known as the debranching enzyme. The debranching enzyme, the mechanism of action is split in 2 phases. So it has transferase activity, and as well as it will have debranching activity. So the debranching activity, you can just simply say it is the alpha-1,6 glycolytic activity. So for the debranching enzyme, it will be this guy here. So again, 2 functions. So this is the first function. So suppose it has a hand, it will come attach itself here. So for the transference activity, it will transfer this here to here. So what does it mean when it transfers here? So it means, again, the first enzyme that we have, the glycogen phosphorylase, again will be able to throw this here at the non-reducing side. Again, cleaving, again this whole process going on, again yielding either glucose 6 phosphate or free glucose. But it doesn't end here. So like I said, it is in 2 activities. So the second activity, because we've left out this guy, he's the last guy to be involved in the whole process. So the second activity is that it will come here, it will break down the alpha 1 to 6 bond. But when it breaks down this alpha 1 to 6 bond, what you yield is not glucose 1 phosphate, but free glucose. And this marks the end of glycogenolysis. I hope it made sense for you guys. If you have any questions, you can come through and ask. If you have any suggestions, or if you want to learn something else, whether it be biochemistry, anatomy, even as far as grade 12, guys, we're here for you. We'll create the content that you need. And make sure that you turn on the notification button. Turn on the bell as well, so that you may receive the notification first as soon as we release more content. We have more plans, more ideas coming for you, and you need to stick around. The best is yet to come.